begin, I have the wonderful honor of being able to introduce Hana today. Hana Sahly is a professor in the Department of Virology and Microbiology at Baylor. And she has a very interesting career. After a very distinguished undergraduate performance at the American University in Beirut, she also completed her medical school training there, where she graduated with distinction. She then matriculated to the University of Connecticut for her internal medicine residency and training, where she served as a chief resident in medicine. She then decided to pursue a career in infectious diseases, and she began her fellowship training at Baylor College of Medicine. When she completed her fellowship, for a short time, she was on the faculty of the Department of Pathology, but then in 2004, she joined the Department of Molecular Virology and Microbiology and Medicine, and she rapidly rose in the ranks to an associate and full professor. I also understand and have read and heard that she is an excellent doctor and delivers really good care. A lot of her care is to patients with HIV. Her research is she will describe it today, but she has been a real leader in the VTU. She also has a national prominence in that she is the chair of the VRPAC of the FDA, and also has been on a number of important special emphasis and steering committees with the NIH. And she's also been exceedingly busy with the Moderna trial, and she will talk to you today about that. And I'm just delighted to introduce her. Hana?
Thank you, Kathy. I just want to point out that I'm still associate professor. The meeting is this month, and if I talk to you guys next month, I'll let you know.
Okay, well, I guess that I just knew, though, that you would be promoted, so I just said it anyway.
Okay, I'll let them know what you said. Okay, so good afternoon, everyone. Today, I will be walking you through the highlights of the vaccine efficacy clinical trial protocol development, the key elements of the protocol development, what went through our minds, what were the questions that we had to answer, and what were the pros and cons of some of the approaches that we could have taken but did not. As we walk through, I tried to keep it down to 45 slides, understanding that it's an average of 1 minute per slide. So we can leave 10 to 15 minutes at the end for your questions. Having said that, please feel free to ask throughout the lecture. This is a small group, and it's good to build the rapport even that way. I'm gonna try to share the screen now. Here we go. Can you see my screen now?
Yes, we can see.
Yeah, we certainly can.
Okay. So vaccine development is in two big stages. There's the preclinical stage, in which the vaccine of interest is tested in preclinical models, and then the clinical stage, where the vaccine's safety and efficacy and immunogenicity are tested in humans, and more specifically in populations of interest. The reality is that the majority of vaccines that are in the preclinical space never make it to the clinical arena. There are various criteria as to what vaccine get moved for testing into humans. Some of these criteria include their potential impact on human health. There are a lot of vaccines that are tested in animal models, but you read about them and they're relatively obscure. The disease incidence is too small to warrant large-scale vaccine development. So the potential impact of the particular pathogen on human health is always a consideration. the safety and toxicity data in the animal model, animal immunogenicity, and manufacturing and release specifications that can potentially meet regulatory criteria. And that is also another hurdle in vaccine development that probably we don't have time to discuss much here. Once in the human testing phase, the clinical phase, the clinical trials are divided into phase 1, phase 2, and phase 3. Briefly to go over the phases, in phase 1 clinical trial, a vaccine of interest is tested in small numbers. An average number is 20 per group, but give or take, usually in healthy young adults, avoiding extremes of age, and patients with underlying comorbidities. And these adults are usually at low risk of acquiring the infection of interest, because it is unclear whether if we give the vaccine to these individuals, whether they develop worse outcome when exposed, and that has happened a few times in the past. The primary endpoint is usually safety and tolerability, and attempting to find the right dose to move into further testing. So they are considered dose ranging, dose finding. Often they are open label, but they can be randomized. And sometimes they are taken further into development under the phase 1 umbrella in a group of interest. And I can think of the malaria vaccines. The phase 1 are often begun here in the United States, if the manufacturer is here, of course. And a small scale study is done. A safe and immunogenic dose is identified in a phase 1 study. And then you move it to the area of interest, for example, Africa, for malaria, or for example, we have a vaccine for schistosomiasis that got moved to Brazil to do phase 1B, because the responses, the immunologic responses and the safety of a vaccine may not be the same in individuals who have underlying high seroprevalence for the pathogen of interest. Assuming all goes well and the phase 1 clinical trial data are reassuring in terms of safety and immunogenicity, a vaccine is moved into the phase 2 step, where a larger number of individuals are enrolled, averaging 50 to 300, which is a number we see often. It provides preliminary information about the vaccine's immunogenicity. So now the numbers are larger. There are men, women, but still relatively small numbers. Safety and tolerability data are better characterized. And the effect of sex, ethnicity, and age, at least in a preliminary fashion, can be acquired, data on these variables. The interval and the dosing are optimized usually during the phase 2 space, and there have been a push in certain cases to have phase 2 followed immediately by 2B or 2/2B, where certain efficacy endpoints are collected. That means usually the sample size is probably not going to be 50 to 300. It will be larger in a phase 2B, but it allows at least to gain insight into whether the efficacy signal is in the right direction. Phase 2A usually tests the efficacy of a particular vaccine in a challenge model, and that is sort of an up and coming. It's been done previously, of course, especially with malaria and other pathogens, but within the VTUs, this is likely to become a sort of a priority way of testing. Which brings us to the phase 3 clinical trials. And these are usually the pivotal trials that are needed to grant approval and licensing of a particular product against a specific indication. It usually assesses the protective efficacy and safety in thousands of people. They're usually randomized, controlled, and blinded, and that is to minimize biases in collecting these data and giving reassurance about the differences between groups. And a vaccine that reaches phase 3 clinical testing usually has specific manufacturing and batch release specifications that are met to allow the regulatory agencies to take these data into account as they review the whole portfolio. The endpoint, again, is efficacy, but it can be a single endpoint or a composite endpoint. But the primary endpoint is key. And that is because the manufacturer or the sponsor usually will request the indication for a particular vaccine based on their primary endpoint. Which fast forward to COVID-19. These phases that I just described to you, from the preclinical to the clinical phases, take usually somewhere in the 10 to 15 years in terms of time frame between the design, the submission of IND, the progression of the phases in a sequential way, 1 to 2 to 3, and the submission of the BLA and the review by the FDA. So 15 years is not out of the range of the expected, but in a pandemic as deadly as the one we are facing, time is of the essence. And the only way that the timeline can be shortened is if some of these steps can be done in parallel. And that's precisely what happened. The phase 1 was begun the injection took place in March, the first injection at the site of Dr. Jackson in Seattle. The phase generated very important data I will share with you that enabled the phase 2, which began in May. But the phase 3 planning, Kathy probably can remember, had its first meeting to lay down the protocol development phase was in April. So a lot of the steps were done at risk, meaning we developed a phase 3 protocol. We did not begin the study, but we spent time and effort beginning the protocol and all the ancillary activities pending the data from the phase 1 and 2 completion. And it could have all been for nothing if, for example, a significant safety signal was observed in the phase 1, or the animal data actually was one of the things we were waiting on. If the animal data revealed enhanced disease, we would have to reconfigure some of the way we collect data on the phase 3. So this was an around-the-clock kind of work done by a lot of groups, trying to align all these priorities and data together so that the phase 3 launches in time with the data required, having developed a lot of it at risk. As we develop a phase 3 clinical trial, and it's not just COVID, the first thing we need to know is we need to know the disease presentation. What I'm discussing here, I want you to think about it in terms of something totally unrelated, like chikungunya or Zika or malaria. The disease comes in different presentation. And to stick to COVID, we have a very large base of infected persons, but who have no or minimal disease. Then there are individuals with symptomatic disease, but not necessarily sick enough to be hospitalized. So it's more a flu-like illness. And then there are the hospitalized persons And these are the persons with severe disease, potentially around 15% of the symptomatic. And then those with critical disease, these are the persons who unfortunately end up in the intensive care unit or die as a result of COVID. And these, depending on what you're reading, anywhere 3 to 5% of those admitted to the hospital actually lately. And they are often older and with underlying comorbidities. So how big is the pool of the uninfected persons? Actually, I had to look it up just for this talk, and it's all over the place. The estimated proportion is very variable, and I found a meta-analysis that placed it at 20%-ish of all infections. Having said that, there are large-scale seroprevalence studies in the United States and abroad that are underway right now. We are even part of one. It's a co-VPN study here, and hopefully we will have information on the seroprevalence that is more indicative of the pool of infected but asymptomatic persons. So why the S protein for the COVID-19 vaccines? A lot of it, again, was built on data and work that was done by others during the SARS and the MERS corona epidemic research response. It was found that the S protein is the primary target of the neutralizing antibodies. Passive transfer and vaccines against the S protein will protect non-human primates from the challenge. There are responses against other proteins, but they are not protective, and in certain animal models may enhance disease, although that was demonstrated with SARS, but not SARS-CoV-2. And in SARS-CoV-2 pandemic, we knew that humans developed robust neutralizing antibodies against the S, and specifically its receptor binding domain. So we knew we were on the right track with the S. The phase 1 clinical trial, we were developing the phase 3 study, so we had to rely on the data from the phase 1 clinical trial. It was performed by Dr. Jackson. In that study, 3 doses were used, 25, 100, and 250 micrograms. And reactogenicity and safety data and binding and neutralizing antibody data were generated. Here we see the issue of the dose selection. The 25 microgram individuals tolerated the vaccine quite well. A fraction of them had systemic or local symptoms, but the majority were mild to moderate. The 100 microgram, you can argue all of them had something after dose 2, but again, the majority was in the mild to moderate intensity. The 250 microgram, you can see that a good fraction of them, although it wasn't a lot of people here, had severe reactogenicity, and I think one person, actually young with no comorbidities, ended up with the emergency room with symptoms. So this is how it was decided that the 250 microgram was not to be moved forward for testing. Here's the immunogenicity data. So the 250 we know cannot be moved forward because of safety concerns. So that left us with the 25 and 100. Looking at the immunogenicity data, here we have the receptor binding domain antibodies, and here are the antibodies against the full length protein. The far right of each panel are convalescent sera antibody titers against these particular antigens. You can see that after 2 doses of 25 versus 100, here 25 versus 100, there is an increase in the immunogenicity, reaching here higher levels than the convalescent as a mean. And this is how the 100 microgram was selected to move forward. As I mentioned, after the promising data from the phase 1, the phase 2 was launched and the protocol development for the phase 3 began at risk. The NIAID networks at the time also coalesced into a COVID-19 prevention network to form the phase 3 clinical trials testing, to perform the trial testing and the monoclonal testing. So at the time, to share some of the background information, we developed a protocol synopsis at DMID. Then I developed a protocol synopsis with the HVTN, with HIV Prevention Network, with whom we merged. And then it was realized that the Operation Warp Speed indicated that since the IND is with the company is in charge of the protocol. So we developed yet another protocol synopsis with the company. So as Kathy mentioned, we had to write and rewrite so many times what we're doing.
I think maybe Martin Becker had a question, right, Martin?
Yes, please.
Okay.
Yes, thank you for that. A question about convalescent levels of antibodies. Is there a specific timing that's defined in terms of after the disease? When are those measured?
These were actually collected. My understanding is that they were 4 to 6 weeks after the illness.
Okay.
And only a fraction of them were hospitalized. I am blanking on how many were outpatient and how many were hospitalized.
Okay. Do I remember hearing right? Is Moderna or somebody considering looking whether a lower dose than the 100 is similarly, protective, or looking more into a lower dose again, or am I making that up?
You are not making it up. Actually, they presented it during their during their VRPAC meeting.
Okay.
Preliminary data on the 50 micrograms, which was not only tested in the phase 2. These data are now accepted for publications. We are not part of the study, but I know that soon it will be made public. They basically found no difference in immunogenicity between 50 and 100. But when we developed and launched the study, we did not have data on 50. So having said that, when they made those decisions, the issue of the variance was not there. So we do know that a slightly higher immune response usually provides breadth, usually. So does 50 microgram versus 100 microgram make a difference when you're testing against variants? In flu, it usually does make a difference, but we cannot extrapolate without seeing those data. And I don't know that we have those data. So that may be a potential benefit of sticking with the 100, at least for now, until we know better.
Thank you.
You're welcome. So we began designing the phase 3 clinical trial, and instead of just going down the route of what is obvious, which is a randomized controlled trials at the individual level, I want to walk you with what are the options if we are testing a vaccine to consider for in terms of clinical trial designs. Individual randomized controlled trials, meaning at each site, each of those bubbles is a site within which half the people are vaccine, half the people are placebo. Sometimes you see a 2-to-1 randomization, but regardless, the randomization is at the individual level. These are usually the most statistically efficient ways to get an answer. The randomization is at the subject level within the site, and it is of value when there is heterogeneity in disease incidence, and that is definitely true when we started designing this study. Not the whole, the country was very heterogeneous in terms of who's having high COVID incidence and who was not. And one thing to keep in mind, is that if the vaccine has an indirect effect, then we can see a reduction in disease incidence in the comparator arms, which may impact efficacy assessment. So there are pros, maybe a minor con. The other way to test a vaccine is if you have more than one candidate, is to do a multi-arm trial. This is also very efficient because within each site, you can randomize to vaccine 1, vaccine 2, and then the placebo will work for both. It is a resource-saving approach and a potential to diversify the vaccine candidates reaching the market faster, and minimizing the effects of temporal trends in disease epidemiology on vaccine efficacy estimates. The other option is the parallel cluster randomized. Here, all subjects within a cluster are randomized and they receive either vaccine or the comparator, usually placebo. Randomization occurs at a unit level, meaning either the unit is maybe a household or a high-risk community or even a town. Small towns in Africa, sometimes they randomize them to one intervention versus another. Or you can also use the ring approach using the transmission network to randomize. It allows the measurement of the total, which is the direct and indirect effect of vaccine efficacy. It is less efficient than the randomized controlled trial, and efficiency was of the essence during this protocol development. Another approach that statisticians like to talk about is the stepped wedge cluster randomized clinical trial. Here, the vaccine is given to everyone, but it's given at different time points for different groups. And that design was highly considered during the Ebola outbreaks because you want to give everyone the vaccine. There was preliminary data that it was working and it's such a deadly disease. But how do you give it and yet generate useful data? That, for example, in that scenario, the stepped wedge approach could be utilized. Its drawback is that it's slow to perform and it's rather complex in terms of planning and analysis. So arguably, since 6 constructs were moving into phase 3 testing, and we knew that kind of by April, May, who are the manufacturers that are furthest along, a multi-arm randomized controlled trial would have been the most appropriate or efficient way to testing the vaccine efficacy. But that would require collaboration and planning between various pharmaceutical. There's also variability in terms of the readiness of some of the constructs to launch within the similar time frame. So it ended up being individualized randomized clinical trials. And these are the 6 products that at the time were considered furthest along. 5 of them are in phase 3 testing. Sanofi met some roadblocks in terms of testing, and the phase 3 clinical trial is a bit delayed. So that's why for the pandemic purposes, the IRCT was the way to go. So IRCT, so that's the design that would generate the data needed in a most efficient and scientifically sound option. But what are we trying to prevent? A study that prevents all the endpoints that are of public health importance is likely just not feasible. There are 3 phases or 3 clinical manifestations of COVID. There's infection with no or minimal symptoms, there's disease, and then there's severe disease that lands individuals in the hospital or give them death. So which one are we to choose? Believe it or not, there were 3 versions of this protocol. To share some background information, there were 3 versions of the synopsis written by different groups targeting each of those. It took so much back and forth, so much discussion to land on what ended up being the outcome of interest. So let's examine the pros and cons of picking these outcomes. And anything that I mentioned here can potentially be extrapolated to other diseases. For example, Zika. The outcome we're trying to prevent is fetal malformation and developmental neurodevelopmental delays in the baby, or neurodevelopmental disease in the baby. So we're not trying to prevent Zika infection. So all of these considerations that went into thinking about the phase 3 for COVID apply to other diseases, in that it's rarely obvious what the endpoint should be. So it will require some discussion. So what if infection as an endpoint? Detecting infection with and without symptoms requires frequent sampling and surveillance. Seroconversion as an endpoint can be a proxy, but it also requires validated tests. Many vaccines of public health importance do not prevent infection, but they prevent disease, or they prevent disease to a larger degree than they prevent infection. So if we put infection as the primary endpoint, we may be setting up the vaccine for failure and not necessarily helping public health. Severe disease. What about severe disease? Severe disease is the most clinically meaningful outcome that we're trying to prevent. But it has a lower frequency, which means we need even larger sample sizes. For most vaccines, preventing mild disease does also prevent severe disease. But we will also, I want to caution you here that it's something that at least should be assessed in the study, because there is history in certain vaccine resulting in worsening or more severe disease in those who got the vaccine. So at a minimum, we should capture severe disease endpoint and compare them between the two study groups. So this is where we landed: symptomatic COVID as the endpoint, virologically confirmed symptomatic disease, which represents a disease outcome of interest. It has precedent with other viruses. This is usually the endpoint that was used for rotavirus, for flu studies. Virologically confirmed symptomatic disease. Also, it has more feasibility in terms of sample size consideration. Another compromise would be to lump disease and severe disease and call it burden of disease, which weighs severe cases statistically more than the mild cases by giving each severe case a score of 2, every mild case a score of 1. And looking at that, I think some of the clinical trial, phase 3 clinical trial, adopted this as a secondary endpoint. But nonetheless, symptomatic COVID-19 was the endpoint. So this is where we landed. The vaccine efficacy was to prevent the first occurrence of COVID-19 at 14 days after the second dose. And COVID-19 was defined by 2 of fever, chills, myalgia, headache, sore throat, new olfactory and taste disorders, or 1 if the patient has cough, shortness of breath, evidence of pneumonia. With RT-PCR positive, most of it is nasopharyngeal, but if they were admitted to the hospital and they got, for example, sputum or BAL, that's positive by RT-PCR, that is also considered positive. But for this and any other trial, the issue of stratification needs to be considered. And we stratify usually on pre-specified variables to account for certain variables that affect the outcome in a predictable fashion. And it often ends up being, in vaccine clinical trials, it ends up being age. More so here because age is such a strong determinant of the outcome in COVID, that the study is stratified by age under 65 and no risk of severe disease, age of 65 and older, or those with presence of severe disease risk criteria, and this included advanced lung and heart disease, diabetes, HIV. Yeah, I think these were the ones. We had to take actually the older ones that were on the CDC website because they changed after May or June, but during the protocol development, these were the ones that were specified by the CDC. Other important endpoints were the assessment of safety in terms of reactogenicity, adverse events, serious adverse events, and imbalance in COVID-19 or severe COVID-19s between groups. This was an event-driven protocol. With a one-to-one randomization, we needed 151 cases of COVID-19 to provide 90% power to detect 60% reduction in the hazard rate, rejecting a lower bound of 30%. And we assumed an incidence of 0.75% in placebo over 6 months. Assuming 15% were to be dropped out, we arrived at the sample size of 30,000. This is the overall study design. Individuals are screened, consented and screened. We obtained history. If they met eligibility criteria, they were stratified, randomized 1-to-1, given 2 doses of vaccine separated by 28 days, and followed for up to 25 months, and they are still in follow-up. If a volunteer is confirmed to be infected with SARS-CoV-2, they undergo daily follow-up for symptom severity. Remember, symptom severity and imbalance between group is an important thing to follow. And they get baseline and convalescent serum collection, and they get every other day saliva for SARS-CoV-2 PCR to determine shedding and viral load differences between vaccine and placebo. The study launched 27th of July. The first site was a CRO site somewhere in Atlanta, somewhere in Georgia, not Atlanta. And one principle in clinical trials is that the study population should represent the vaccine target population. One problem we faced is that the clinical trial participation is traditionally predominantly Caucasian, and that was just how it is in most studies. But COVID-19 disproportionately affects minorities. They have higher incidence of disease, higher mortality of disease. And this is how it began. So this is 27th of July, first enrollment. In red are white study subjects. You can see that the study, when it began, was overwhelmingly Caucasian. And we had to implement changes. Somewhere around here, it was determined that this is just not acceptable, and that enrollment of non-whites has to be prioritized. So the study is representative of the population in whom the vaccine will be given and who will stand to benefit the most actually from the vaccine. So we had to turn the tide. Initially, there wasn't a lot of buy-in from the company, but it took commitment from Operation Warp Speed leadership and beyond, actually. The DSMBs pre-specified that diversity is a study metric that has to be followed. Co-VPN operations were mobilized. The co-VPN registry was rolled initially only to co-VPN sites, but then to improve the diversity across the study, the co-VPN registry was rolled out to all the sites, the private and the academic. And they helped us with educational and promotional material production. The co-VPN leadership reached out. Dr. Fauci and the Surgeon General came to the study teams a few times, and met with the PIs and indicated the need to keep our eye on the goal of enrolling minorities. Moderna agreed to the consensus and decided to stop enrollment at certain sites that were predominantly enrolling Caucasians and allow the sites where there's diversity to contribute more to the study in order to diversify the study population. And the sites understood this and they started using the registry preferentially. They sorted their registry by race and ethnicity. Many local community leaders in terms of churches and whatnot were recruited to help the studies, and we started giving interviews to local media that is catering more to minorities, whether it be Latino radios or TV and African American, preferentially African American stations. The study did, however, from the get-go enroll a good proportions of those who are older than 65. You can see 25% were older than 65. 17% were young but had risk of severe disease, diabetes, COPD, heart disease. And then 58% were at risk, not at risk of severe disease, but we had to be very selective in terms of choosing individuals who are not telecommuting, individuals who are, if they were young, of course, not telecommuting, who had kids in the house going to school. So they had to have exposure in order to be on the study. And of course, geographic diversity was well represented. We had 99 sites in 31 states. It was a very demanding study, but nonetheless, you know, we managed to stop it almost halfway through, reverse, increase the minority enrollment, and meet the target numbers. So we enrolled a little over 30,000 at the end of the day. It was 79% white, but that includes also the 20% Latino, many of whom identify as white on the race questionnaire. 10% African American, 4% Asians, and more than one race, 2.1%. You can see that these numbers kind of mirror the US population. The vaccine reactogenicity after the first dose, there you can see that it was more, this is in younger, this is in older persons. You can see the difference between the vaccine and placebo in terms of injection site reactogenicity after dose 1. The majority of it was pain. But if we go between here and here, you will realize that the dose 2 was more reactogenic. And also, we still saw the difference between young and old, in that younger had more reacto. Systemic reactogenicity was also more prominent compared to placebo. And see here, the systemic reactogenicity is what's most increased after the second dose, the majority being fatigue, headache, myalgia, and arthralgia. If someone, this is a question I get asked almost every 3 days, sometimes 3 times a day. What about if someone was already seropositive? Do they have more reactions after the vaccine? Actually, they had less. I keep pointing them to this slide, and there was a table in the supplemental in the paper, and that patients who were seropositive at baseline and got the vaccine not knowing they've had COVID before actually had numerically less reactogenicity than those who were seronegative, and that is systemic and local. So what did the vaccine do? The vaccine overall was very efficacious. Oh, one thing actually, we needed 151 cases, like we saw in the analysis. And after all the diversification and all these efforts, it ended up that the sites that contributed a lot to the cases were in the Wisconsin, Dakota areas, because this is where the outbreak that took place in the October-November timeframe. Nonetheless, it was since then there have been many more cases representative of older individuals and minorities, but it was just a little background information here. So the vaccine was 94% efficacious at preventing COVID-19 of any severity after a median follow-up of 2 months. You can see here that the vaccine efficacy was in the 80s and 90s in all subgroups, whether it's young or old, or real old, but here there were only 7 cases, men or women, those with comorbidities, and those who are communities of color or non-Hispanic whites. In terms of severe cases, this was a very important data to collect. We had 30 severe cases, and the efficacy against severe cases was 100% because there was 30 in the placebo, 0 in the vaccine. Since then, we know actually right after the data cutoff, there was 1 case here in the vaccine. But nonetheless, the study continues to accrue cases, and the next data cut is going to be real interesting. So now what? The vaccine is efficacious, but there are many more questions that need to be answered. We need to detect less common or delayed safety events. For example, will there be enhanced disease severity as antibody levels wane? Everybody remains in follow-up for up to 2 years. The durability of the vaccine efficacy is an open question, and we are gathering data pertaining to that. The correlate of protection is unknown, and that is also under intense research activity. Efficacy against emerging strains was a non-issue up until a few weeks ago. Now we know that sera from the vaccine recipients have a five-fold reduction in neutralizing the South African variants, but that's in a pseudo-neutralization assay. We are awaiting data on viral neutralization assay, and a collaboration was established actually with the South African group just a couple of weeks ago, and we had to facilitate that. And do we need a boost? And if so, which strain should we boost with? And also, this is now a topic for a study within the BTU. The fate of the vaccinees after receiving EUA for the vaccine, emergency use authorization. Those on the vaccine arm, were unblinded, and they are, many of whom opted to stay on the study and continue to give us data on incidence of COVID. And those on the placebo arm, as you can imagine, the overwhelming majority of them wanted the vaccine, and now they are also providing data on the disease incidence. The way we're going to compare this is delayed vaccination versus early vaccination, and compare these 2 arms in the long run. Initially, there was talk of a blinded crossover versus unblinded crossover, but eventually it's the unblinded crossover that was adopted. And I think I reached the end of my slides, trying to put the story as to how this evolved. And I'm happy to take questions now or by email.
What a great summary, Hana. That was great. Questions? Do you have? You can put questions in the chat box, or you can just raise your hand, or you can just ask them. So this opportunity is a wonderful opportunity. So questions.
This is Anthea. So that was wonderful. Thank you so much. So one thing that I found a bit surprising was when you mentioned that Moderna, or that the sponsor was not open to kind of looking at further diversity, and that you all kind of had to push to say, hey, this is really important. Do you think that may change their approach moving forward? And how do you think that we as future researchers can make this become less of an issue moving forward?
I'd like to think that what happens after COVID is not what happens before. There's a line in the sand that was drawn here. There's a principle. A phase 3 vaccine clinical trial needs to mirror the population in whom you are going to give the vaccine. If you're going to give a pertussis vaccine to pregnant women, you cannot go study it in old men, So there is that. So I can see why for phase 1 and 2, if you have a clinical site, let's say, I don't know, in somewhere in the middle of the country, it's not fair to be asked to be representative of the study of the population of the United States, But if it's a phase 3 and it's a vaccine that's going to be given to everyone in the United States, and it's a vaccine that significantly will have a bigger impact in communities of color, then it's imperative that this gets that they get represented in phase 3. Having said that, I mean, not to defend anyone, but the industry was facing 2 pulls: speed, okay, and well, when they started laying down the plans for speed, it was realized that, well, not so fast. So because of this, because of that, so they were being pulled in different directions, and arguably so the fact that they heeded the advice is commendable, and we are grateful for many people who sort of emphasized the need for diversity to them. And we do see how the subsequent clinical trials that came after Moderna are not facing this issue.
Great.
And I think some of it is because of that emphasis early on in the Moderna study.
Yeah,
Thank you.
Hana, I really wanted to commend your effort and the effort of your team in the Moderna study in really insisting that there has to be diversity. And like you said, in all of the subsequent study and currently the Novavax study that is recruiting, that is not an issue at all. We track the data and the diversity is just impressive. And from day one, they have kind of built it into the protocol that they have to recruit people from underrepresented minority and people that are at the highest risk for this pandemic. So thank you so much, Hana. That was really, it was a great precedent that you all set, and thanks for this very impressive story that you have told.
Thank you.
I think the other point that is important to make is that it would be helpful if the investigators looked like the people that they were recruiting. And so when I first became involved in a lot of this, most of the investigators were men. Now, obviously, that's not the case, and so we need to make those same strides in investigators of different ethnic and racial groups.
Yes, and that starts very early on.
Yes.
So,
and that is why I'm really encouraged by the diversity of our mentee, the group that we now have pulled together. It's really exciting to see how diverse this group is. This is the future of the consortium or the network.
Yeah, that's great. One thing that didn't come across is, if I can give an advice, is be friends with your statistician. I always find that I learn so much from, like, they get drawn, they're mathematicians, right? Like they need to be pulled into clinical reality by our interactions, like what you bring to the table. But you learn so much from them by intensifying the frequency of interactions with them as you go along in protocol development. And so don't get intimidated by how dry it is. It's, you know, if you interact with them, it becomes more accessible, and it will give you a new light on interpreting your own data.
So Hana, can you share with us how you came to, how did you get here to become the chair of this very important protocol at this very important time?
Oh. Well, ask Kathy and David Stevens. I don't know.
Maybe I can answer that. I think that Hana always does her homework. Hana always is thoughtful. She has spoken and articulated her concerns all along. I listened to her when she just started. And all of these things are ways to get to where Hana is by doing your homework, by volunteering to do something hard, by doing it well, and by just, you know, just doing the right thing, and you do get rewarded for your hard work.
Yeah, thank you. Thank you all. Please email me with your questions and ideas. And
thank you so much for your time for the presentation. This was great to hear it straight from you. We do appreciate your time. I see there's a couple of hands up. So
bye, everyone.